Right, so basically, a summary of cellular communication is that receptors are protein molecules inside of a target cell or on the surface that receives a chemical signal. Chemical signals are released by signaling cells in the form of some soluble molecules called ligands. Ligand binds another specific molecule, delivering a signal in the process. Ligands and receptors exist in several varieties. However, a specific ligand will have a specific receptor that typically only binds with that ligand. And there's two different types of receptors, internal receptors and cell surface receptors.
Internal receptors are found in the cytoplasm of the cell and respond to ligands that cross the cell membrane into the cell. Cell surface receptors are found on the cell membrane. They bind to the ligands that do not cross the cell membrane. Small hydrophobic ligands can pass directly through the cell membrane. They typically interact with internal receptors. Water-soluble hydrophilic ligands cannot pass directly through the cell membrane.
Once a ligand bonds to a receptor, the signal is transmitted through the membrane and into the cytoplasm. Continuation of a signal in this manner is called signal transduction. Signal transduction only occurs with cell surface receptors because the internal receptors are able to interact directly with DNA in the nucleus to initiate protein synthesis. Signal transduction pathways can be extremely complicated and involve a large number of enzymes and other proteins.
Kinases are a type of enzyme that adds to a phosphate group to another molecule, including other proteins. This is called phosphorylation. Phosphorylation can activate or deactivate other proteins. Second messengers are a small group of molecules that help spread the signal through cytoplasm after a ligand bonds to a receptor. They do this by altering the behavior of certain cellular proteins.
A class of drugs that leads to distortions of reality and perceptions are known as hallucinogens.
LSD, also known as lysergic acid dithylamide, is a hallucinogen, a serotonergic agonist, as well as a dopamine. LSD's activity is located at a subtype of serotonin receptor known as the 5-HT2A and is critical for the drug's psychedelic effect on the body.
This receptor is located on the surface of the neuron membrane. A neuron, or nerve cell, is different from a normal cell because it communicates through an electrochemical process and is longer, thus taking more time for the signal to be received.
The space between LSD and the membrane is called a synapse. Thus, this signaling is known as synaptic signaling. Protein folding makes travel distance slower, making it easier for receptor proteins to receive the synaptic signal. LSD has an extremely high affinity for the serotonin 2A receptor and is remarkably potent.
To say that hallucinogens stimulate serotonin 2A receptors means that they mimic the action of serotonin at the receptor by binding to it, altering its shape, and ultimately altering the internal conditions, and therefore the behavior of the neuron it sits on. Neurons are the nerve cells that help send and receive signals that are produced by the LSD.
By accepting more serotonin, the receiver gets high by producing more signals for the nerve cells to receive, producing more serotonin in essence. Ultimately, LSD changing receptors allow them to activate at a higher rate.
This signal, transductor, and receptor trio requires millions of signals, which requires a lot of ATP. Since there's so much phosphorus required in this synaptic signaling, enzymes and cellular cycles are crucial. Enzymes are used to decrease the requirements to produce these signals, and cycles like glycolysis, Krebs, and the ETC are able to produce the ATP needed.
In addition to the actions at the serotonin receptors, LSD has a high affinity for dopamine receptors.
Because LSD is a unique ligand that can simultaneously bond at the serotonin and dopamine receptor subtypes, it is an excellent tool for probing the biochemical basis for behavior. One large-scale effect of LSD on the brain is due to increased glutamate transmission in the frontal cortex.
The GABA-A system, also known as the dopamine neurotransmitter system, regulates cortical arousal and when stimulated produces sedation.
This dopamine system modulates attentional and goal-directed behaviors and enhances alertness. LSD also increases heart rate rapidly, which means more chances for molecules to pass through it. Repeated administration of LSD over several days promotes rapid tolerance for the drug and has much less of an effect than initially did. This is associated with the downregulation or decreasing number of 5-HT2A serotonin receptors. This is why LSD is not considered to be an addictive drug.
As LSD travels and interacts with the serotonin 2A receptor, the protein folds over the LSD, creating a lid, trapping the LSD. Your brain responds by taking in the receptor to degrade the LSD, but this degradation can take almost 12 hours to happen.
Researchers found that the drug causes parts of your brain to communicate in unique ways, especially in the visual cortex, which can cause you to have vivid and complex hallucinations.
The different expressed genes after acute LSD treatment encode for a variety of proteins.
These genes include immediate early genes, which are trigger genes in the genome.
One example of these IGs includes CFOS, which encodes for a nuclear transcription factor that in turn activates multiple downstream genes. The CFOS gene is rapidly upregulated by many stimulatory pathways and is often used as a marker to determine general neuron activation. It suggests that LSD activates many brain areas.
Another nuclear transcription factor gene induced by LSD is CROX20,
which encodes for a zinc finger transcription factor.
Basically, CROX20 decides whether CFOS gets transcribed or not.
The ones used to treat ADHD, like Adderall and Ritalin, are classified as stimulant drugs.
Stimulants are both harmful and helpful at the same time. To prevent overstimulation, the body has transporters that move their residual transmitters back to the presynaptic terminal. When someone has ADHD, their brain doesn't have enough dopamine or norepinephrine to bind to the receptors on their postsynaptic terminal, so they look for other stimulation sources. This is what amphetamine salts, Adderall, and methylphenidate, Ritalin, which are two of the most commonly used stimulants to treat ADHD, help with.
Share symptoms of constantly being hyper, active, or inattentive. This is not their fault, however. This is caused by the lack of dopamine and norepinephrine in their brain. Dopamine is a neurotransmitter that is associated with the brain's reward center, and norepinephrine is associated with memory and alertness. Typically, a person without ADHD has dopamine and norepinephrine in the presynaptic terminal in the neuron that are then shot in the synapse and bind to their respective terminals on the postsynaptic terminal.
Dopamine receptors are mainly found in the coronary, renal, and cerebral tissues. When stimulated, vasodilation decreases vascular resistance in these areas.
When certain parts of the brain are exposed to too much dopamine, for instance right after the individual takes an illicit drug, other behaviors may be present.
These can include aggression, hallucinations, twitching, nausea, vomiting, and depression.
Dopamine is a neurotransmitter that regulates mood, enhances pleasure, behaviors, motivation, and attention. Tyrosine is transported through a sodium-dependent tyrosine transporter. Once in the neuron, the tyrosine gets hydroxylated by the enzyme known as tyrosine hydroxylase to L-DOPA, and this is converted to dopamine by the aromatic amino acid decarboxylase. Then it is transported to the synaptic vesicle where the enzyme dopamine beta hydroxylase converts the dopamine to norepinephrine. Calcium then floods the neuron, which causes the vesicle to fuse with the membrane and release its content into the synapse. Finally, the norepinephrine binds to the postsynaptic receptor on the effector membrane, which then triggers the intracellular response.
Adderall is made up of dextroamphetamine and levophetamine, which are two isomers of the amphetamine molecules. Its structure also reveals a benzene aromatic ring bonded with the amine side group. Ritalin is made out of methylphenidate that also consists of hydrogen, carbon, and nitrogen. Adderall works by boosting the overall amount of dopamine and norepinephrine in the brain. However, Ritalin takes a process of transporters that prevent overstimulation and cancels it out, leaving the dopamine or norepinephrine to bind to their terminals on the postsynaptic terminals.
Ritalin targets the dopamine transporter in the prefrontal cortex area of the brain. The main action of modulation for dopamine and norepinephrine. A norepinephrine/dopamine reuptake inhibitor blocks DAT, which inhibits the dopamine from being completely reabsorbed in the synapse. Inhibiting DAT stops a presynaptic neuron from the reabsorption of neurotransmitters.
Adderall blocks the reuptake of norepinephrine, dopamine, and serotonin into the presynaptic neuron and increases the release of these monoamines into the synaptic space. Adderall also enters the presynaptic neuron, building a membrane potential to make dopamine come out of the storage vesicles and expel them out towards the synapse. Adderall also has the ability to reduce the effects of the enzyme monoamine oxidase.